Dr. Joe Miller. Okay, well, let's start again. I'm very happy to welcome Dr. Joe Miller, who is the GBIF Executive Secretary and who came from Copenhagen to us to have him here to give a presentation. Dr. Miller got his PhD in 1997 from the University of Wisconsin-Madison, then followed at CSIRO with a postdoc in Australia, working on acacia phylogeny, then was senior research scientist at the University of Iowa, where he built a genomic laboratory, and then moved back to Canberra and was a research group leader at the National Herbarium in Canberra, working on phylogenetics and biogeography. This is followed by a 5-year tenure at the NSF, first as program officer and then as the head of the international science diplomacy efforts in biology, the NSF program. Since 2019, he is the executive secretary of the Global Biodiversity Information Facility in Copenhagen. And as a researcher, Joe has worked on evolution, biogeography, and systematics of Australian plants, particularly the genus Acacia. Because it's Australian acacia, it's still acacia. And he's also known as Acacia Joe on X, formerly known as Twitter. And his talk today is entitled GBIF's Infrastructure Enables More Natural History Museum Impact. Joe, please.
Thanks very much. It's great to be back in the Midwest. I'm a local. I grew up in South Dakota, so I took advantage of this trip and visited two of my brothers in South Dakota over the weekend. And I also did my undergraduate work at the University of Minnesota, so I'm all Big Ten, so it's great to be back in the middle of the Big Ten here. But today I'm going to focus on GBIF as an infrastructure, and my talk is really focused on these people right here, the G13 science leads who are here. But I'm sure the message will resonate broadly, or at least I hope. So what is GBIF? The Global Biodiversity Information Facility is an international network and data infrastructure aimed at providing anyone, anywhere, open access data about life on Earth. So we're an intergovernmental organization. We're not part of the UN. We're separate. We're unique in that way. And we are funded by voluntary contributions by governments. And those countries that voluntarily contribute have a say in our work plan and government, or my boss, and I respond to them. So that includes the United States, UK, Belgium, Denmark, Germany, France, and Netherlands. I say those countries because we have people represented, and Canada represented here. All really foundational members of GBIF and very important in our work. However, we are located in one of those. We are located in Copenhagen, which I'm very delighted to say I live and work there, especially in the summer. It's a wonderful place to live. We are hosted by the government of Denmark for the last 22 years, and we are co-located in the Natural History Museum at the University of Copenhagen, and represented here today by Michael Story. So we have a great relationship with the museum, and we're all looking very much forward to the opening of a new museum in Copenhagen in the next couple of years around this beautiful botanical gardens within central Copenhagen. We get a lot of great resources and collaboration from the university. It's been a great relationship for these 22 years. They really help us and subsidize our infrastructure, which is no small task. So we really appreciate their collaboration. But what we really are is a network of people and data. And over 22 years, we have built a infrastructure to share data that people like you share from your museums and other sources, and people like you use for your research and policy initiatives. Currently, data comes from 136 countries, and we have, I believe it is 4 new institutions publishing new data sets every week. So we are definitely a growing network, and you can see we still have some weak spots there in North Africa and the Middle East. And we are trying to have an in-person meeting in Jordan, but that's really hard to plan given global events. We're really trying to fill in the gap of having data providers all over the world. That is our mandate. The G is global in GBIF. And the foundational part of GBIF is that there are really great natural historians in every country of the world. And it's actually not that hard to get those people connected to each other. Once you start getting people in the same room, Russians and Chinese and South Africans and Brazilians, we all love Americans, we all love biodiversity, and it's a common language, and it's really quite easy to get people to share data. It's just making that connection to the governments in order to get national membership is the tricky part for us, but I think you can imagine why that might be. So we do like to talk about numbers. We currently have over 2.6 billion occurrences in GBIF. So this is evidence of a particular species at a particular place at a particular time. We have 64 country participants in 43 organizations. 43 organizations include organizations like I Think Bio in the US or DISCO in CCAF in Europe. We also have a growing number of publishers, almost 2,200 publishers, nearing 100,000 different data sets. And I'll talk about this in a couple slides, but nearly 10,000 peer-reviewed publications that we can track to particular data sets. This number here is crazy, if it is true. The entire data set of GBIF is downloaded on average 3 times a day. So it's a very high throughput infrastructure. So, billions of occurrences coming in and out at every day. And on average, 5 new occurrences come in every second to GBIF. So we are a very large infrastructure. All right, so I'm just going to give you a brief overview of how we provide evidence for research and policy. When I say we during this talk, that means everybody in this room. It just doesn't mean GBIF Secretariat, because GBIF Secretariat is nothing without the data. I mean the data providers and the data users and our of course, the governments that want and ask GBIF to do this. So this slide shows 3 different parts of the evidence for research and policy, and I'm going to walk you through some of these and kind of tell you a little bit about the history of GBIF. So originally, 22 years ago, when we started in Copenhagen, the foundational data sets of GBIF were natural history collections, like the Field Museum. This is, of course, and still remains the most robust data because it's all connected to a voucher. So this was kind of the first epic of GBIF data. Second epic of GBIF data was probably about 8 to 10 years ago when citizen science data come on strong. So we have data from groups like eBird, which is our single largest data set, iNaturalist, Observation International, which is a big citizen science player in the Netherlands and Europe. So this is kind of the second epic of data. But now we're in the third epic, which is very exciting, and we're kind of calling this the machine-derived data epic. So the first data flows from this are camera traps, and we are working very closely to make camera trap data available. But also eDNA, environmental DNA, is part of this. And this is part of our strategic plan going forward. We are able to use open licenses and standards developed by community. And the community means people that work in your museums who are developing these standards. And I'll talk about that in a bit. Our infrastructure is very robust. We bring in data through something we call the IPT into our global index, which I'll talk about. Then we provide monthly snapshots on cloud services. So if you want to run GBIF data against satellite imagery in Amazon, you can do that. It's all right there waiting for you. The biggest access point for data is through the API. We're getting pings all the time for access to data through our packages and things like that. And we have other kind of curated ways of getting data from GBIF through hosted portals, which I'll talk about. And we are in the process of completely updating our portal from into this new hosted portal format, which I will be showing you. I do want to emphasize this is that how we transform this data, your data, into something that's used by people. On average, on the next slide, I'll show you there are 5 peer-reviewed publications of GBIF-mediated data every day. So I imagine every day there's a new publication that is using data from the Field Museum. I would say, I'd never check this, but it's probably 2 or 3 publications a day that are people that have used data downloaded from GBIF or publishing it. This probably, could be people having nothing to do with the Field Museum. Because what we do with the power of GBIF is that we're able to combine different data sets into a single data set. So if, for example, if you wanted to find out all about the red oaks in North America, instead of going to 100 different data sets, you can come to GBIF and we take the red oaks from 100 different data sets, put them into a single data set for you with standards, put a DOI on that and send it to you for your use. But this is important because in this day and age, we all want our data to be used not just for our research, but for a larger good. We have a very close relationship with the Convention of Biological Diversity, IMBES. We're on committees in there, behind the back of the room, saying data, data, data. Don't forget the data in all these procedures. And we do have, every publication that comes out of GBIF, we do correlate them to different thematic areas such as invasive species and conservation. And this slide shows the growth in GBIF mediated data over time, expecting over 1,700 publications in 2023. I don't think I have the final number for that. I need to update this slide. But the growth is growing, and our scientists are using the DOIs. However, not all scientists use the DOIs from the downloads, and we have a full-time staff member who tracks through the literature to find uses of GBIF data so that we can provide that evidence of use for you. So what I want to talk about today is kind of maybe a simplistic way of what GBIF is. Our head of informatics, Tim Robertson, talks about it as a data lake, and this is a speak from the informatics world. But what GBIF is a place where you can share your data, and as a field museum and individual museums, you share your specimen data with GBIF, thank you very much, through standardized methods. But you're also sharing your metadata on your collections as well. How many collections you have, who's the manager of the collections, what is how many, for example, even the Latin long of your collections. So metadata about the collections. And what we do as an infrastructure is we apply data models and use the standards that your communities are developing. So research scientists from the Field Museum and other museums are very active in these communities of TAGWIG, the Biodiversity Information Standards groups, to make these standards that we all use. And it's very important. And the more time I spend at GBIF, the more I realize this. So what we do is that we take this data from this portal and we can mix and match it in many different ways into portals and dashboards. And that's what I want to show you today. And this is made possible through some improvements in our infrastructure recently. And of course, the data in GBIF is just not those three balls. We also have observations, environmental DNA, camera trap literature, many different forms that I showed you from a couple slides ago. But most of the day, I want to talk about the Global Registry of Scientific Collections. So this, as the name cites, is an attempt to get a database of all the scientific collections in the world. This is a project that we took on about 5 years ago. Completely wrote the GR Cycle database, which was basically a database of metadata about collections. We rewrote the infrastructure and have been having a series of community consultations about this over time. And right now in this GR Cycle database, we have evidence of over 7,398 institutions, 7,860 collections, and now that number is over 210 million specimens. So what we've done is take this infrastructure, which is a metadata about the collections, then we have the data that you give to us in data sets in regulargbif.org, and we've combined them so that your specimen data now is attached to your collection, which is probably, for most institutions, a better way of thinking about your data. So this has been a big advancement, we hope, and I'm going to walk you through what that means in the next few slides. So the portal, we actually christened it at our governing board meeting in Australia back in October. This new interface for it, this is the institution page of GR Cycle, and don't do it now, but the URL is gbif.org/scientific-collections. So this is just a map showing the distribution of all the institutions in gbif.org now. See not very many in Greenland and Siberia, but really lots of collections all over the world. It's really fantastic we've been able to take input data sets from many different places, such as the single largest data set in here is Next Herbariorum, the well-curated list of herbaria. But you can see here, there's a row for the metadata for each of the institutions, and we can click on those and get more information, such as here for the Field Museum. This is the information that you shared with us about the collections, about the institution of the Field Museum. And what we've been able to infer from that is that 43% of the specimens that you shared with us are from the US. That you have approximately 40 million specimens, and we have evidence of 2.2 million shared. And these are the institutional disciplines that you have described to us within the collection, the institutional page. So this is a view of a collection page. So if I go back up here, let's see. I forgot to show here. The Field Museum, we have evidence that you've told us there are 20 separate collections within the Field Museum. That's how you have organized your institution. So we drill down to one of those collections, this time the NHM in London, the herbarium there, and we have more detailed data because it's a smaller entity and we can be more precise about our descriptions, taxonomic descriptions and use terms, et cetera, worldwide geographic distribution. And we have the number of specimens shared with GBIF and some important collectors. So we just have been able to pull this information from the data that you have shared with us. And this is on the About page. And I'm going to walk you through the specimens and dashboard page of this collection view of GBIF. But first of all, from this, we're also able to display on this page within GR Cycle of the nearly 1.1 million species specimens in GBIF. These are of nearly 107,000 different species. You've given us that information in all these data sets, and we're able to basically go up and count the number of species. That is, but after we have done all of the standardization to our taxonomy. So this is the specimens page of NHM Herbarium. And you can see here, this is a normal view of our new interface for specimens. Each of those dots will expand out and have multiple subdots into it, and you can drill down into individual specimens. And not surprisingly, you can see in bright red there, the UK, where the museum is located. And most of the specimens are from there. So what I want to spend the next few slides on are dashboards. So since you have shared your data with us and of the collections, and we know what you have digitized and sent to us as specimens, we can make some simple dashboards about them. These are exemplar dashboards I'm going to show you, but these can be done, other dashboards based on the data you share with us could be created as well. But first of all, we have the metadata again that I just first showed about the NHM Herbarium. And then I'm going to show, I just took a bunch of screenshots of different dashboards within about 20 different metrics that you can find here. And this is all available in the data that you sent to us. We're just putting it in a nice graphical, easy way to view. So this is, again, that same statistic of 106,000 different species. However, nearly 178 taxa. So there are many more, lots of subspecies or subspecies in your data set. And we also can give you some richness metrics of how many fields in the Darwin core are filled out for your collections, how many have Latin longs, how many have country, geolocated, and things like that. And we recognize that this data is hard to have in the collection management system, but we're just displaying what you have sent to us. And things like this, I think, are quite interesting as well. This is a histogram of the event date. So these are when the collections were made for this herbarium. There's one back in 1592. Is that real, or is it's real? All right. So, going back to 1592, those are the ones that are shared with us. There are probably other really old ones that haven't been digitized yet, be coming in So these are the dashboards that we can easily make. We don't just take the data you share with us, we also connect to outside sources such as the IUCN Red List and the IUCN GRIS list, which is the invasive and alien species list. So we know what species are recognized as on the red list and the invasive species list. So are there any red list species in NHM London? Yes, there are. And there are 305 specimens of this Hedlundia angelica, which is a near endangered plant of the family Rosaceae. So by connecting different data types together, we're able to show back to you some of these information that might not be readily available in your own collection management system. And these are the kind of statistics I like. You know, which family has the most representation in the collection? I was surprised to see the Fabaceae at the top here. I thought it would be the Solanaceae or something like that. We have, of course, orchids are second, and Brassicaceae third. And then because each specimen there's an opportunity to put an identifier in the Darwin core field. So we have evidence of that. And not surprisingly, Sandy Knapp of NHM has identified more of the collection that has been shared with GBIF than any other scientist. And she's been a stalwart there for several years. So these are kind of dashboards that can be easily made from the data you share with GBIF. And here's another one. This pie chart isn't really a pie chart. It's just a representation of where the specimens come from. The largest percentage from the UK, then Australia, then India, USA, China, Paraguay, Brazil, Nepal. It's kind of fun to go to collections and see where it's from. I'm a botanist, so I went to Kew Botanical Garden, and at least a third are from Brazil. Maybe it was even a half. So it's kind of interesting you can really understand where their focus has been. All right, so that was kind of an overview of the dashboard. And now this is a specimen record. If you've gone to gbif.org recently, in our current portal, which is being taken over by this portal, there's two columns, one with the data you sent to us and one with the data that we have inferred from that. And sometimes that's updating based on taxonomic synonymy. And sometimes it's just filling in gaps in the records that we were able to infer based on the other data in the system. So this is what the new specimen record looks like. We have this is collected in Chile. However, we do not have a Latin long for this, so it's not, it's a preserved specimen here at the Field Museum and collected in 1961. And this specimen record has all the information that you were able to share with us in the collection management system, such as the summary, and I could scroll down to show you more of these. The summary, the taxon, the location, the occurrence, the event, the organism, the identification, and the citations you share with us. So any data that you share with us, we can automatically put into the specimen page in this new GR cycle attached to your collection. There's something else that's been going on behind the scenes that we decided to make public a few months ago. These are these little clusters or diagrams. So when we index, we are indexing data from multiple types. And I mentioned we have evidence from DNA, from the literature, and from specimens. So we index relevant data from GenBank or ENA. We have millions of material citation records of DNA evidence from GenBank or ENA or Barcode of Life. We also getting to have hundreds of thousands of records from material citations from the literature. So how a specimen has been cited in the literature, that has been harvested from the literature and submitted to GBIF as a data set. So we have different types of data that have some relationship to them. And that isn't always obvious. But what we've been doing for the last about 4 years, is running an algorithm. You can call it AI if you want. But we're looking across data sets for similarities in a scientific name, a date of collection, and a place of collection in type status. So this algorithm runs, we now have it running every week. And we used to call it the clustering algorithm. It gives us these nice clusters, but we kind of changed the name to related records now. So these are hypotheses of related records in the collection. And I'm going to go through a couple of these examples and go through those colors. So this is a cluster that I just circled here. The kind of the mustard color is the specimen itself, and the one with the browner out of point is the entry point for this cluster based on what I clicked on in the data set. This cluster only has red and blue balls, small ones. That's telling us that this individual specimen, each of these specimens is a type specimen. So you told us that this is a type specimen, and we have 4 of these 6 specimens have an image. So this is pretty good evidence that this is and this is actually a senna, a plant, and us botanists, we like to do this for a type. We'll collect multiple branches, press them, and send them to all of our friends around the world. And then we lose track of them, right? But if that data is all shared in GBIF, we can bring that data back together. And perhaps we can learn from, this is the same tree. Perhaps we don't have to sequence this thing 6 times if it's all the exact same organism. So we could have, but we don't have any evidence that this one has been. So this is Senna obliqua. This is from NHM London. Here's evidence from a data set from the Royal Botanical Gardens Kew. And this one here is from Munich. So this was collected in different duplicates sent out to at least these 6 different herbaria. And we're able to bring that data back together. And I'll give another example in the next section of this talk. So as a summary, GBIF is able to connect and visualize various types of data. So the richer the data, the more complete the data you send to us, we're able to link it and connect it and show it in various ways. And GR Cycle is a new way of displaying that away from just gbif.org, everything thrown together, or just within your individual data sets is how you share data to GBIF. We're bringing them into hopefully develop a holistic view of collections. And that's what GR Cycle is all about. So, but what about the undigitized collections, right? We know that the majority are not digitized. Can this framework be used for the undigitized collections? Yes, it can. And we've been working with you for several years on this, on being able to make some information about the quantities of collections that are undigitized into GR Cycle. And this really is, this GBIF's work on this has really come out of the Global Collections paper that came out 1.5 years ago. And we're trying to implement some of those recommendations that came out of that paper so that you can understand where gaps are in the collections that could be focused on for digitization. So we have what we call a GR Cycle roadmap. This is a 2-year roadmap. And the first year we developed this new user interface that I just showed you. This user interface is for GR Cycle itself, but it also can be used for any subset of data in GR Cycle, such as a national collections portal, which I'll show you, or even an individual museum. So these 13 museums all have robust data structures and web pages. You may not need a collections page. However, a small university collection may not have a way to visualize their own collections. If they share the data with GBIF, we can share that back to them through a hosted portal. So we've developed a mechanism for community updates. And probably the most important, most recent thing is that we have the community, the Tegwin community is reviewing the data schema around Latimer Core, which is the collections descriptor standards. And this is now being implemented, and we are implementing it within GBIF in collaboration with you in GR Cycle. However, in the next year, we are focusing on the structured collection descriptors, which relates to the Latimer Core and institutional surveys. And institutional surveys is what directly came out of the One World Collections paper. So what are structured supports? What are structured collection descriptors? So this is the work of the Latimer Core Working Group. It allows us to generate, you can generate descriptors of what's in your collection, such as I have 1,000 birds from Thailand. In my collection, we have 100 grasshoppers from South Dakota. Now, if you have that detailed information, even though it's not digitized, that kind of description data could be sent to us and made part of this robust data set. Most likely, you don't have that detailed descriptors, but it gives you a framework for sharing those type of data. And this is really what I'm here to do today, is to ask you to utilize this resource, and let's really get a good data set here. We have an infrastructure to hold this, and it's flexible, and let's see if we can really make really good data sets out of this. So this is going to be a big focus of the next 4 months. And we're going to be reaching out to the community with webinars and how to implement these Latimer core standards. We really love to have the G13 be the leaders in implementing this with us. And this is what it looks like. The tables, for example, you have an institution, Acme Museum of Natural History, have an insect collection. You can have a descriptor for the Diptera inventory, giving how many male and female flies of a certain species. If you have that information, or more simple, just that you have an estimated amount of a particular genus of arachnid. So this is only doable to the level of information that you have about your undigitized collections. But it's the detail that you have, if you can share that with these descriptors with us, we'll be able to aggregate that across different museums, just like we're able to aggregate what you've digitized, we can aggregate what you haven't digitized, and then we can compare across those. Which brings us to the institutional surveys. So the G13 led a large survey of, was it 87 museums around the world, with particular questions about what's in their collection. So this is coming at it not from the really granular level of the species, but at the top level. Because we recognize, and I learned more this morning, each museum is very different, and it's very difficult to have robust connections across museums because you're all arranged very differently. So we have this Latimer core view from the bottom up with the details that you have, but we also need something from the top down with a very high level. We have 10 million angiosperms, things like that. And that's the institutional survey. So the G13 and the One World Collections made this survey a few years ago. We will be archiving those survey in a machine readable format. Those won't be editable because they were a survey, but certainly if there's another survey coming on, we can certainly add those as well. And our hope is that there are multiple surveys out there that the community can merge on a single survey format. And this could really help us to understand and compare, contrast across collections. And these, as I mentioned, these institutional surveys can drive dashboards. So we have dashboards I showed you on the specimens. I can't show you the dashboards on the undigitized collections because we don't have that information in Latimer core yet. But it's very exciting, the opportunities we have now to put the data you do have into Latimer core. We'll put it in GeoCycle, and then we'll, together with your help, we'll come up with dashboards to look at digitization. For example, how far do we have to go? How many more? If we have 700 million plant specimens in the world, we have 100 million in GBIF. That means we have 600 million left to go. And then we have a target. And the granularity of how far we'll be able to go depends entirely on the data that you're able to share with us and our ability to link those together through the Latimer core standards. But this is a very exciting prospect for us, and also hopefully will be helpful in your museum management as well, and outreach to potential funders. So this is kind of what it all comes down to, is it's customized hosted portals that not only contain what I showed you, the digitized collections, but information about your undigitized collections. Complete, and we could even, and I have made a hosted portal of all 13 of your museums. And it's quite simple to aggregate this data at any level as possible. And a G13 one, a CTAF one, IDIG-Bio one, a US one, but it's all possible. It depends on how you slice and dice the data. So this is, I think I'd like to put this out as a kind of a challenge to us all, is to make use of this, of the data that you have, share it with us over the next 6 months with these Latimer core standards and institutional surveys. And let's see how far we can take these customizable host portals to display how far we are in the digitization project, and see where the major gaps are. It's not going to be simple, but I think this is kind of this is the sort of thing that GBIF was built to do, and we can't do it without your data. So there are some really great examples of this, and it's actually the UK has been pushing us on this, coming up with great ideas, asking us to implement for their UK National Science Collections portal, and coming up with ideas on dashboards and some ideas on the citation tracking as well, and better showing that. Obviously, lots of interest as well from the Smithsonian, helping us out financially with developing GR cycle through this next part, really seeing the value of this global resource. And I say global because I'm very proud of this next slide. I did have a slight connection to Ecuador in my life. And Ecuador is our most recent hosted portal. Did you know that there are 56 natural history collections in Ecuador? That's what the GBIF node in Ecuador tells us, because they have been curating this data set for the last year, and now have transformed it into a data portal. This is all driven by GBIF data, but all the language, the text, and everything, of course, is in Spanish on the other pages in this. But it's their own, patrimony is the natural history collections in Ecuador, and this is their way to talk about that. And this is available to any country that's a member of GBIF, any institution, whether you're in a country or not that's part of GBIF. We'd love to have a portal for you if you would like one. All right, I would like to shift over now to the data model work, which is very well connected to this. So the data that you share with us now is based on the current data model of Darwin Core. What is it, where is it, who saw it, and when was it seen? This is a very complicated system. It worked very well for GBIF in the beginning days, but it's not able to share the richness of the data that's in our collections. So the past few years, in the next several years, we are moving towards an improved data model. And I'm going to kind of explain that by another bubble diagram here. So this is a bubble diagram of specimens that are shared in GBIF. And what this is showing is not only, before I showed the type specimens and images, but this cluster also has information about sequences, because we have occurrences in GBIF from GenBank and from IBO. So this is the specimen from the Cal Academy. It was sequenced from the Cal Academy by Barcode of Life. They took a picture of it here, and that's the specimen record in GBIF. We also have a material citation. So somebody wrote a paper based on the specimen, and that data set in the paper was submitted to us. So we know that somebody used this specimen. This is actually a specimen. I think this was published in PLOS One about this very specimen. It was a species revision. And here is the occurrence based on the CON gene that was sequenced. I'm not sure if this one is the eyeball one or it's a separate one that was sequenced by a research group. So basically, through this clustering mechanisms, we have this knowledge of related records. The data model is bringing all that together and putting it in a single specimen page linked to your collections. So that specimen page in GR cycle will be much richer. That's our goal. And this is built around an event-based data model. In there in blue, there was an event at a particular location. The event had a protocol. The event interacted with an organism. The organism had a determination of its name. All of these are different things that happen in our day-to-day work as a field biologist. And this information now can be robustly captured in a data model that can be represented in GBIF and other places. Here's an example of this. This is an old one. I kind of like this example. It's from, it was collected in around 1900 in Benin. This is an old specimen like that. It doesn't have a lot of information about it. We don't have a particular locality other than in Togo, sorry, not Benin. But it does have an image here. And the collection hypothesized that it might be a type. So this is what the data model looks for that particular specimen. It's not very rich. We had an individual at a site in Togo. There was an occurrence event. We know this person, Kirsten, collected it, made a specimen. Later, somebody did a photo of it, and then somebody called Voigt came along, changed its name, and wrote a paper. That's in the data model, right? We could infer that not too hard by just looking at this specimen. However, I found a data set kind of randomly, and this is actually the first part of the story, but I've retrofitted it to make a story out of it. I found a data set published when I was on X of a nice paper that took measurements of about 200 species, 200 specimens for 29 characters, and they published it and did some PCA analysis of this, straight on, nice morphometrics. And the data set for that was in the supplementary files of the paper. Wonderful, best practices, but no one's ever going to use that data set, right? So what we did is that we took that data set and we published that data set to GBIF because we knew that there were records in that data set that had exactly the same barcode or herbarium record sheet as that specimen I just told you. So somebody took that 130-year-old specimen and measured it and published it. But those two things were never going to be connected. But if it was all published to GBIF and it had that identifier, we could link them. So this is what it looks like now. I just mentioned all of that. We also have these three people, Xanthos, Mayo, and Leridan in Europe. They took that specimen, they made some assertions of measurements, and they published a paper about that. So we have two different data sets about the same specimen, and now we are going to link those together in a single specimen page. So this is not a very, so this is what the specimen will look like in GR cycle. It has all of the data information normally that came in from the Berlin collection, but we have a citation here now of the Xanthos and Mayo paper because we know that the specimen was used in this paper. And here, if we scroll down, there are all the measurements that were in that paper as well. So this is a flattened basal sheath persistency. That's what they measured, and this is the trait, and it's connected to the specimen. It was published to GBIF. And here's another one that is a bit more robust of a collection from University of Kansas. In this case, a collection management system has a lot of data to it. Normally we couldn't put all of this data in GBIF with the normal Darwin core. We'd have to use we simply couldn't do it. But using the new data model, we're able to show all of the robust data come from that collection management system, including all the DNA sequences of it, 12 images, and a length assertion of that fish. So all that data was not shared with GBIF before. With the new data model, we can do that. And one other thing that I'm quite proud of is that we do now have one phylogenetic tree published to GBIF. This is I think a lack in our community. There is no fair digital repository for phylogenies anymore. So we have been working closely with Treebase and Open Tree of Life. And on the side, I've started to publish phylogenetic trees. And this includes all the specimen data for this tree. So this is a small tree with 31 specimens in it, but we have the metadata for the specimens, and we'll be able to link to each specimen this phylogeny. And this is kind of just the first stage of this. And the next stage is that our friends at Q are publishing a plant and fungal tree of life soon with 8,000 genera in it, and they're going to publish that citation to us, so we'll be able to link that phylogeny to hopefully, well, as many set of specimens as possible that they use in that tree. So it's showing the value of your collections by publishing other data sets. So this is kind of what we're doing in the digital extended specimen space. We're trying to help the community by linking these different types of data to the original specimen, field notes, morphometric data, images, sequences, descriptions, phylogenies. We're doing what we can to make the data model robust enough so that if you publish data that has this type of information in it, we can attach it to the specimen. All right, I just have two slides that are probably I hope to be the most impactful part of our work over the past couple of years and over the next year is a thing called Checklist Bank. This has been a long collaboration between Catalog of Life and GBIF. Catalog of Life is an aggregator of taxonomy and is the resource of most of the taxonomic names in GBIF, but not all, maybe about 80% of the names. And we've been working very closely with them to get a better joint infrastructure to share names so that we can make a better GBIF taxonomic backbone for aligning all of our occurrences too. And this will greatly improve the data quality of GBIF. The taxonomic complaints is the single biggest complaint about GBIF data. Of over half of the notifications we get through the help desk are about taxonomic complaints. And we don't currently have a good system to do something about that. with its collaboration, a catalog of life. We're building that system together and including ways to reach out to taxonomy communities, more easily share their taxonomies into checklist banks so that we can get into this global taxonomic checklist. So this is what we've done with catalog of life. So on top is how they made their global checklist. They managed several different import data sets such as IDAS and worms and world flora online. And these are all vetted, curated data sets, and that's the foundation of catalog of life. What we've done is we've built infrastructure that allows us to, with machines, add on other data sets to that. And these are the data sets that we need in order to put that last 20% of biodiversity into our taxonomic backbone. And these are in what we call the extended release of the catalog of life, which we will be, over the next few months, we'll be releasing this as a GBIF taxonomic backbone. We have one now, and it's in the testing stage, and we're developing quality control, quality assurance protocols, because we really want to make this an impactful data set. And it does include not only the core catalog of life, it includes smaller data sets, but it also includes publications that we derived from the literature that have been harvested via PLASI of some of those data sets. So if one of your researchers publishes a new species, that is very likely the same day that that's published paper hits the airwaves, there'll be a data set in GBIF that will go into checklist bank so that we can incorporate it into the catalog of life. That's kind of the big goal here. The data that's out there, bring it in as quickly and high quality as possible so that it can be reused. So what does this mean? What does checklist bank do? Currently, it's an open repository for taxonomic checklists. And we have some very good tools for comparing across checklists. We have a lot of work to do in communication and front-end development. It's a bit overwhelming, but there's a lot of opportunities here. And that's why we're taking our time to have the release of this. And we really think this new GBIF taxonomic backbone will have a large impact. In order to facilitate this, GBIF and Catalog of Life have decided to come organizationally even more closely related to each other. So GBIF now serves as the administrative host of Species 2000, which is the parent aid group of Catalog of Life. And this allows us to work more closely together in this joint project on the Catalog of Life. We don't know what this is going to lead, but it's starting a discussion of how Catalog of Life and GBIF perhaps will integrate, perhaps will stay separate. We don't know. The original vision of GBIF included something like Catalog of Life to be part of GBIF. If that would have happened 22 years ago, we'd be in a much better place now. But the originators of GBIF decided there wasn't enough money to be able to do everything that was asked of it. So we went along the road of occurrences, and taxonomy was left out. So now we're playing with the idea of bringing it back in because it's really absolutely critical to everything we do and everything that you do. All right, so what's next? A couple of concluding slides here. What's next for collections at GBIF? I hope I've convinced you that GR Cycle is worth your time and effort to mobilize data and edit. So we'll be implementing collection descriptors and institutional surveys over the next year, and having more community engagement. And perhaps there could be a specific working group of the G13 leads in GR Cycle to meet with our lead on this, Marie. So we really can get a really foundational set of high quality data from your museums as examples for the rest of the community. The data model is going to continue. We're currently working on publishing models for ecological data and interaction data. And we have starting to build out the catalog of services, which includes this material specimen catalog, which is my title for the specimens within GR Cycle. Of course, we're doing a few other things. Things that we would like to do is improve the citation tracking, citation tracking. I'd really like to link this back to funding agencies. We have a little teaser on that, but we'd like to be able to tell the NSF, you paid the Field Museum, you gave the Field Museum $200,000 to digitize these grants. What are the publications that came out of that? We could track that, but we just need to have a few more things in place. And I'm going to be at the NSF in a couple of weeks, and I'm going to pitch that idea to them. Also, DNA flows are going to improve, hopefully with the GBIF. We have a very large ideas in eDNA. eDNA is great, but there really is not a system to combine eDNA data sets that because of the taxonomy. And we have some ideas on how we can build a robust taxonomic set to compare across different eDNA data sets. And we're talking with many partners on this across Europe and North America to, and of course, trying to get money to do this. Ecological interaction data and a phylogenetic tree repository are also on the radar. So here are my requests for you as a community. Please dedicate a staff member to curate your GR cycle data. And let's make a working group and have that interact very closely with GBIF, with Marine. Lead implementation, so that be part of the implementation of these collection descriptors so that we can have a very good example for the community of how to compare across museums. This standards business is very important. Every time I think about it, I go, yeah, what your scientists are doing kind of on the side, going to meetings, TEDWIG and being on these working groups is critical because that is what makes it possible for this Darwin core upgrade, for the Latimer core, for the Humboldt core. Very important that your scientists are engaged in that. Please engage more in the data model development. This is going to go on for decades. Lots of information on trait assertions that can be put into the data model. In that regard, please consider publishing data that's not in your CMS. You have thousands of data sets on different hard drives in this museum and every museum around the world. Our goal is to be able to find mechanisms to get those shared so they can be attached to your specimen. That's a kind of a long-term goal, but that could also be kind of a pilot project as well. And then traits, trees, and taxonomies. Please make sure you share those data sets. That's all I have. Thank you very much for your attention.
Yeah, thanks a lot for your talk.
Thank you.
I'm sure there are a number of questions, and we have microphones on the left and right hand side. So.
That's a great, exciting talk, Joe. So I'm a curator of insects here at the Field Museum, and especially excited about the aspects of the undigitized part of the collection, which, or most of our collection, it will be for some time. When I was looking at your talk, immediately, and you're showing the new module with all the collections, and there are the collection IDs and codes, I immediately thought about taxonomic publications. All of them list the collections that were used for some work and where the specimens came from, even when the specimens don't have an ID or a barcode or something. Is there any thought of tapping into that kind of information to get some idea of all the undigitized part of the collection?
So I mentioned at the beginning of the talk, there are about 95,000 data sets in GBIF. About over half of those are from one organization called Plazi. And what they do is they harvest data sets from the published literature, and you get data, sometimes multiple data sets every day from them. So if there's a scientific publication, a revision of an insect, they're going to harvest the material cited section of that. If you have your specimen data in there, there will be an occurrence in GBIF called a material citation occurrence that has that specimen record in it. So there's a lot of data like that, and that's really what got me intrigued in all this, is that if we can do that, and then we can tie it to the specimen, and then we can tie other types of data, and we can just bring it all to that specimen. It's quite exciting. So that data from Plazi is fantastic. Machine-derived data sets can be tricky. So we have to work on the QA/QC part of that. That's part of what we're doing right now with these partners. And then we also, BHL obviously is another great resource of data, and there are lots of some ideas of harvesting more data sets from those digitized BHL papers.
We do have a question from the chat. What's the difference between COL and the Encyclopedia of Life? Which should I use?
Catalog of Life is a catalog of taxonomic names, a vetted taxonomy, so accepted names. Encyclopedia of Life is more of an encyclopedia of data about species. I do believe the Encyclopedia of Life uses GBIF, taxonomic backbone, as its taxonomy, but they provide two different resources. One is a vetted taxonomy that's really critical for scientific use. Encyclopedia of Life has this information about individual species, but it also very importantly has data on the, especially the last 5 or 6 years, the trait data is a very robust trait repository of traits generally at the species level, some of at the specimen level as well, but more of at the species level. So think of Encyclopedia of Life as species information and traits about those species. Encyclopedia of Life is really focused on the names. Part of our ecosystem of organizations.
So good talk. I didn't know all that about GBIF. I have 3 questions, all of them are pretty easy. First of all, what is that crustacean up there?
Excuse me?
Just faded white.
What is?
The crustacean up there? The
I
red,
have no idea.
Oh.
Somebody told me it was there's gotta be a curator around here.
that one, yeah.
Hermit crab?
That's what I thought it was. Okay.
Okay.
So next question. How long did it take for specimens to become digitized and then hit GBIF?
So as long as, after they are digitized, the current process is that a Darwin Core archive is created using the standards of Darwin Core.
Okay.
That is published to something called an IPT, which we have literally hundreds of these instances around the world. And I'm sure you have one here at the Field Museum. It gets published to that Darwin, and we're constantly, what's the word? We're browsing all the IPTs. If a data set hits the IPT at the Field Museum, we'll pick it up within a few hours. And then we run it through some QC analysis and it'll go right into the database. So once it's ready and you push the button here to put it on the IPT, we will pick it up.
Okay, so almost immediately sounds like.
Yeah.
Final question. I'm a taxonomy nerd. What can I do to help?
Yes, we need taxonomic data sets. And we need, so we're kind of in a very interesting phase now where we are get this new infrastructure into a format in which we can use it in GBIF. So we're really internally focused on getting this infrastructure to work on GBIF's backbone. And there's a lot of development work that needs to be done for that. But in the process, we also need to build out the mechanism so that if you have a data set, I can specifically say what you need to do to get that shared. And that is very, very high on our list. But it's not as high as getting this updated in the GBIF taxonomic backbone. So we have 2 editors that work on the taxonomic editing, this extended catalog, and those would be the contact people to send data sets to. You can publish a data set to catalog of the checklist bank, and then that would go into the pool of data sets to be used. That would be the first thing to be able to do. And the editors are looking for specific data sets to fill specific holes. So I can give you, checklistbank.org, and you can publish data sets directly to there.
Hi, thank you very much for your lecture. Your talk was very inspiring. And I am not from here, I'm from Brazil. And I would like to know what the plans are for fostering community engagement. So not only here in the US, but across the globe in terms of thinking about institutions and collections that are not yet listed and engaged with GBIF.
Yeah, thank you for that. So I didn't go into a lot of details, but we have a national node in any participant country. Our national node in Brazil is very active and highly producing node. I visited them in August. And also in Brasilia, we have ACTO, the Amazon Cooperation Treaty Organization, which is a associate participant, which is working across the Amazonian countries. And between the 2 of them, we have a lot of a hope for building out some data mobilization projects. As far as Brazil goes, Brazil is kind of the focus of a lot of the activities around the Global Biodiversity Framework Fund. So there's going to be data flowing to Brazil for biodiversity, as it should be for the Amazon. And this is from our conversations with, from the CBD, et cetera. So one of our focal points is foci for our networking area is to build a data mobilization. We have a data mobilization program called BID that we ran 6 million euros over a few years, but it wasn't in the Amazon because our funders didn't, it wasn't a priority. But we're specifically have our eyes on some money that would fund data mobilization in the Amazon countries. And I'm pretty sure that that's gonna happen in the next few months, and that can really build out that needed data mobilization for Amazon. However, of course, we need that everywhere, and that's the longer goal. But I'd be glad to put you in touch with our node in Brasilia. We actually have a contractor, full-time contractor based out of Brasilia as well. And she's working on a vector of human disease project, mobilizing data the vectors of human diseases, mainly focusing on malaria. So we do have a good footprint in Brazil.
Okay, thanks a
Thank
lot,
you.
Joe, for this interesting talk and the discussion, and thanks to everyone for coming. Thank you.
Thank you.